Hello, and welcome to the Patient-Centric Sampling Interest Group webinar for this month. This month we have Christoph Stowe from the University of Ghent presenting for us on microsampling. What do we need to do to accelerate real-time uptake? But before we get started, just a few notes for everyone. We'd like to thank our partners, the Evidence Base, who help us host these webinars, and of course our sponsors for the Patient-Centric Sampling Interest Group, without whom none of this would be able to happen. Just so you know, our agenda today is, I'm doing the introduction. I'm Neil Spooner, the chair and co-founder of the Patient-Centric Sampling Interest Group. Christoph will then do his presentation for about 40 minutes or so, which will be followed by a Q&A. If you can use the chat function to submit your questions, please, and then I will be able to ask Christoph those questions at the end of his webinar. Please note that the webinar is being recorded and will be uploaded and accessible from both the Evidence Base and the Patient-Centric Sampling Interest Group website after this event. And after it all, I'd just like to do a quick wrap up and have the opportunity to tell you a little bit about the Patient-Centric Sampling Interest Group and point you towards where you can find more information. So with that, as I say, for the Q&A, please use the private chat function, and the webinar is being recorded. And with that, I'll disappear and hand over to Christoph.
Okay, thank you, Neil. Try to share my screen. Hold on a second. Yeah, that should be it, I guess. We'll go to the beginning. Could you confirm you see everything well, Neil?
Yes, Christoph, it's come through.
Okay,
yeah.
Okay, thank you.
Fine.
Okay, thank you for introducing me. It's a pleasure being here, presenting at the Evidence Base webinar today. And actually, while preparing this presentation, I thought, okay, how will I find a somewhat different angle to introduce the topic? And one of the things that I thought about was how would AI define, let's say, accelerating the implementation or the uptake of dry blood spots. So I searched Bing AI for description of accelerating a dry blood spots and being taken up by a researcher. And this is what I ended up with what's depicted over here. So indeed, the topic of today will be microsampling, and more particularly, what can we as researchers or as community do to accelerate the real life uptake. Actually, while preparing this presentation, I stumbled across a recent publication just a few weeks ago, published in the Journal of the American Chemical Society, about X-ray and infrared microanalysis of Mona Lisa's ground layer and the significance regarding Leonardo da Vinci's palette. So now you may wonder, am I in the wrong webinar? Am I in the wrong seminar? Did I miss something? No, you're right. You're in the right one. Actually, if we look at the abstract of this particular paper, in the abstract, already the first words deal with microsampling. And it reads, an exceptional microsample from the ground layer of Leonardo da Vinci's Mona Lisa was analyzed by high angular and so on and so on. So actually, we are dealing with microsampling here in this scenario. And more particularly, if you look at the Mona Lisa and you focus on the hands, and you look in very much detail to the middle finger, and I give you all a few seconds to really focus on what you may see there. Do you see it? There's a tiny drop of blood hanging there, a microsample. No, I'm just kidding. It's not really a microsample there. Actually, there is a commonality between microsampling in arts and in patients, but it's not about the fingertip of the Mona Lisa. In a sense, what the authors of the GACS paper did and what we do can both be considered as some kind of centric sampling. On the one hand, painting-centric sampling, on the other hand, patient-centric sampling. And if we look at the Mona Lisa, obviously we could say that nobody wants to be responsible for ruining the Mona Lisa. So obviously in this context, this painting-centric sampling does not imply sampling at the center of the painting, and also obviously not in the fingertip here, but at the site where actually we can get relevant information about what we actually want to deduce from this painting. What about the ground layer, the paints, the mixtures that have been used, and so on, which are those things that were used by Leonardo da Vinci in the early 16th century. Likewise, if we consider patient-centric sampling, why would we want a patient to give a macro sample, a tube of blood, if actually we can get the same answer to our question with just a single drop of blood? Why would we want a patient to travel 5, 10, 20, 50, or 100 kilometers to just give a blood sample for just a blood draw? Why would we want an immunosuppressed patient to come to a hospital or to a doctor's office amidst coughing and sniffing patients if this immunosuppressed patient can just provide his or her sample from the home environment? Something else which is in common between the painting, Mona Lisa painting, and patients is that they both represent a precious source. I guess we can all agree that the Mona Lisa is rather precious. I looked it up, and roughly already back in 1962, it was estimated at a price of 100 million euro. If we take inflation into account, that would account to something like roughly 1 billion these days. Also, patients are precious. Think about the elderly, but also think about children. And in those instances, a minimal invasive sampling, like a finger or arm prick, can be really beneficial. Also, for instance, when thinking about laboratory animals, also there, microsampling confines to the 3R principle, representing a refinement in technique and also leading to a reduction in the number of animals used, and actually even with better data quality. Obviously, we are also interested in having a representative and a good quality sample. Of course, we do not just want any sample. In the case of the Mona Lisa, we want a sample that does give us an answer on the question, more particularly what sampling was performed, or what technique was performed by Leonardo da Vinci. And that's why we need to get a representative sample. And in this case, the representative sample was here in the very upper right corner of the painting. And like that, by sampling over there, we could get relevant information, or the authors could get relevant information about the technique that was used and the paint that was used. Likewise, we can only get we can only expect a good result from a good quality sample also if we consider microsampling. In a sense, that's where everything starts. And also when considering a representative sample, that's actually what we really want. We want a blood sample or a serum or plasma sample derived thereof. And because we actually know what a serum or a plasma concentration means, we can also use a blood sample. And it shouldn't matter whether that's a regular blood sample or a microsample from a blood context. Talking about microsample, we are confronted with a limited amount of material. In the context of the Mona Lisa, obviously, we don't want to scrape half of the Mona Lisa surface to get an answer to the question that we are asking. Likewise, we don't want, and importantly, we typically don't need, in many instances, a whole tube of blood. Think about newborns, for instance, or think about patients in which we need really frequent sampling. Hence, microsampling can really be considered an advantage. However, at the same moment, also microsampling may pose a disadvantage or a challenge, as it's really a micro sample, with only a very limited amount of material being available. To make things concrete, if, for instance, we consider a dry blood spot, which is collected on regular filter paper, the same which is used in a newborn screening, And we only take a 3 millimeter punch from that, as is conventionally done. If we then only consider 1 nanogram per milliliter concentration of a certain analyte, that means that this 3 millimeter punch would roughly only contain in total 3 picogram of analyte. Typically, if we proceed with the sample preparation, if we proceed with the injection into a system, we can't collect everything on the one hand. We can't have a recovery of 100%, and we can't inject everything. So it could be that we inject only 1 picogram or even less on the instrument. And obviously, this does pose a challenge in terms of sensitivity. And there we come to a next important point. We need sensitive techniques. In order to get new insights from the painting of the Mona Lisa, new high-end techniques were required. Likewise, the advancements of analytical instrumentation made it possible in real life to determine minute quantities of analytes which are present in a microsample. Think about these sub-picogram amounts of material which I was just referring to. So, in a sense, these advancements in analytical techniques have really made it possible to move the field forward from the conventional newborn screening field, where high analyte concentrations were present, to newer fields. Think about therapeutic drug monitoring, biomarker monitoring, where much lower analyte concentrations are present. And obviously, we also need a trustworthy methodology. I already referred to the fact that we need good quality samples, but equally important is the availability of a trustworthy methodology. Only when using the right sample and the right trustworthy technique, we can get to trustworthy results. This holds true for the Mona Lisa, but this is also true for dry blood microsamples, for instance. We really should be taking care of the methodology and that it's properly validated so that we are sure about results that we are obtaining. Because these results may determine whether a patient may get a different treatment. And unfortunately, what we know is that many methods have not been firmly shown that they are truly robust to be really applied in real patient practice. So there's still work to be done. And of course, we want to gain new insights. Microsampling of the Mona Lisa has led to unique and new insights into how Leonardo da Vinci prepared his paint. Likewise, microsampling in patients may yield unique new insights. For instance, think about the presence of a micronutrient in a population, for instance, in remote Africa, or about to the extent to which certain population has been exposed to forever chemicals, like, for instance, PFAS in communities near industrial production sites. Or think about, for instance, fluctuation of drug levels if you consider home sampling. And I'm sure the audience can come up with many new scenarios where also microsampling may lead to new insights. And last here, different angles. I'm sure many of you will be aware of the fact that irrespective of the angle at which you look at the Mona Lisa, you will always have the impression that the painting sort of looks you in the eyes. And also microsampling can be looked at from a different perspective. There's also many angles from which you can appreciate the benefits in microsampling. From a clinical perspective, a patient perspective, an epidemiological perspective, even an animal perspective. And obviously, yes, also, and very importantly, from a cost perspective. But also I come back to that later on. So now, after having laid the ground layer from this for this presentation, lead free as opposed to Leonardo da Vinci's paints, it's now time to move on and switch gears to come to the actual topic of this presentation: how to really accelerate the real life uptake. Now I'll come to a bit more scientific approach of this presentation. More specifically, if we consider what can be detected in a dry blood microsample, my answer would be typically sort of everything can be detected in a dry blood microsample. You can look at DNA and mRNA, for instance, considering expression profiling, viral load monitoring, genotyping. These are possibilities. You can look at proteins, For instance, you can consider the enzyme activity, antibody titers. Think about, for instance, antibody titer monitoring during the pandemic. Drugs, there's more and more biologicals which are being marketed, biomarkers, and so on. You can also look at trace elements. Potassium is one example. We've used potassium as a correlate for the hematocrits. Metallic profile, you can look at, for instance, at the wear of a metal-on-metal prosthesis, an application which we have shown to work for cobalt. You can look at lead, for instance, for environmental purposes, and you may think of different examples. And of course, typically, if you think about therapeutic drugs, then we may think about small molecules. Those could be endogenous compounds, as in the case of newborn screening, but also phenotyping, therapeutic drugs, drugs of abuse, doping substances. And so there's many examples that you could think of. So you could think, okay, there's already such a wide variety of applications, such a wide variety of analytes for which dry blood microsampling has been shown to work. So can't we just say, okay, been there, done that, and move on and just start applying it? I would say not really. About 10 years ago, there was a bit of hype, and sometimes there was some over-promising that with dry blood microsampling, we could do sort of everything. But if we look in the clinical community, if we look at the real implementation in routine care, then this is actually quite rare. And so actually this uptake by the real clinical community is really actually slow. So why the hesitance? Why isn't microsampling being applied already very widely in the broader community? Like I already said, there's quite some advantages that are related to microsampling. I won't go over all of these. I just listed them up. Quite often stabilizing effects. They offer convenient transport and storage, simplification of sample preparation, amenable to automation. They also are represented economic procedure, easy and minimally invasive, a small sample volume, and you look at the representative matrix, as I already said. And also, if you think about animal sampling, it confines to the ethical principle of the 3R principle, being a refined procedure and leading to a reduced amount of animals that's being used, for instance, in preclinical studies. But it's not just advantages, there's also several challenges and issues, and several of those have really posed a break for the breakthrough of microsampling. There could be recovery issues, stability issues, volume issues. The sample quality, as I already said, is really important. Spot inhomogeneity may be one issue. Also interpretation issues. So you can look at the blood concentration versus the plasma. Capillary venous differences could be in place. Hematocrit effect. I won't go into too much detail today, but that's something that we've worked on quite heavily. And like I already said, you're confronted with a limited amount of material. So there's quite some things that need still to be tackled. So how to accelerate this implementation in routine care? How can we try to move the field forward? So in other words, how can we accelerate? How can we move from this space to this space? I tried to think about this and listed up some points that I think are quite important. First of all, we need good science and good studies. And quite importantly, we need solid data. Provided an instrumentation in a certain lab is sensitive enough, I typically say that anyone can measure anything in a dried blood microsample. It's not about that. It's actually about whether the measurement is right and whether the methodology is robust. In other words, can you really provide a trustworthy answer to the question? Can you really rely on results to really change, for instance, the treatment of a patient based on this dried blood microsample? To help move the field forward, we and others have worked on a guideline and that provides some guidance on how to establish a robust methodology, because that's actually what's really important, having a robust methodology so you can really rely on the results. Importantly, also how to interpret the results. If you look into literature, then quite often we see that a capillary dry blood sample is compared with a plasma sample, and then a correlation is made, and then people say, okay, we're happy, we find a correlation. But it would be strange if you wouldn't find a correlation between, for instance, a dry blood finger prick sample and a plasma serum sample that corresponds with that. Obviously, you expect a correlation. The question is, can we really translate the result from this dried blood microsample to a serum sample. And that requires quite some more analysis. For instance, you may wish to validate first that if you look at a venous blood sample and you compare the result with the dried blood sample derived thereof, that actually you get the same result. In other words, it shouldn't matter if you look at, for instance, 10 microliter of dried blood or 10 microliter of liquid blood, you should get the same results. And robustness of the extraction is really important for that. Likewise, it may be important to look at the venous plasma ratio, because there could be differences intra or intra-individually, and you could also have variation in the blood plasma ratio. And so this is important to define this, because if there's a large variation between the blood plasma ratio, it may just imply that you cannot just simply translate a blood concentration into a plasma concentration. And you may just wish to work with blood concentrations rather than plasma concentrations. And importantly, then you could also have capillary venous differences. So it's something that you measure in a finger prick sample may just, because of physiological reasons, just yield a different result from if you would use that same sample in a venous context. And there's nothing wrong with that, but it may just imply that you should not use these results interchangeably. And only then, if all of this has been evaluated, then you can really make the comparison between the capillary dried blood sample and a plasma sample derived from a venous blood sample, and say if you can really convert a dried blood sample concentration to a plasma concentration, whether you see a correlation, whether that correlation is good enough, yes or no. So good science and good studies is essential. Also, we need to perform our studies under realistic conditions. In other words, something may work in the lab, but that may not be reality in which the study will be conducted or in which the application will be conducted. We have to wonder, where will the study be conducted? What's the context? Will we have supervised sampling, or, for instance, will we have self-sampling at home? Will patients be able to provide samples that we actually require? What will the transport conditions be? If we have to transport something from the equator, where conditions may be tropical, is our analyte still stable under those conditions? So those are all typical questions that have to be asked and that have to be addressed before really going for an application into the real life situation. And here just one example for a tyrosine kinase inhibitor in which we applied microsampling. And actually what we show here is we look at concentrations on the y-axis, we look at different patients on the x-axis, and the different points that we see represent different samplings for every patient. For instance, here, if you look at the second last patient, patient 18, and then we see, and these are the green triangles, then we see that if the gray zone is actually where we want the patient to be, that on one day the patient was below this gray zone, and on another day where actually there's no reason to expect a different concentration, this patient is above this gray zone. So in other words, if we would sample on day one, we would consider concentration to be too low, and if we consider on day three, we would consider the concentration to be too high. Which one is the correct one? We actually don't know. So we could actually wonder, what's the value of just having a single point measurement, and whether that measurement was from a microsample at home, or whether that measurement was from a single measurement being performed in a clinical lab. We can actually wonder, does a single point measurement tell us the truth? And in a sense, we could consider patients nowadays as pretty much black boxes. We put in a pill, we put in a certain medication. We know that we have many good medications that actually are capable of working in patients, but patients are black boxes. We don't know what the concentrations are, and we can have a single point determination in the clinical lab, but we don't know whether that single point determination is actually really reflecting realistic conditions at home in the home environment. And microsampling may provide an answer to that. Important for microsampling to work, we need motivated patients. Like I already said, we need good quality samples. And good quality samples is one thing, but we also need to think about the patients. We need to think about the patients that have to provide the samples. And so in other words, we need to have a patient-centric approach. We have to make it the patient as easy as possible to provide the sample, just for instance by allowing home sampling, convenient sampling in the home environment, but also by allowing the patients to collect the sample in him or herself in an easy and convenient way. So in other words, we should make sure that the patient is capable of giving us the right sample. And there's several new developments, for instance, that have taken place that allow the sample to collect in a really convenient, really convenient manner a sample in this home environment. And important, it's not just that we should define what the patient should do and that we should be thinking what the patient should do. We should listen to the patient, and also looking at patient groups, having involvement of patient groups, and those patient groups telling us, no, this is what you thought of in your lab environment, and that's just simply not realistic. This is what's realistic for this patient group, and this is what's relevant for this patient group. So we should really open up to those and listen to those. And like I already said, there's several alternative devices that have facilitated dry blood collection. Here's just some examples that are shown. And there's multiple examples that are present, also several ones that are not listed here. But in a sense, it comes down to the fact that multiple approaches have been set up that allow the patients to collect in a trustworthy manner the samples in the home environment. And it is possible to collect trustworthy samples because if we then look and score the sample quality, we can really get good quality samples. Before, we've published a study in which we really looked at the sample quality and actually found out that the sample quality, even from those individuals that were only instructed in a remote manner via leaflets and via online video on YouTube, if you really make sure that these patients have the right incentive and are really motivated, they can provide good quality samples. And also, if you then look at the patient's motivation, it's really important to make the patients co-responsible for their results. In other words, for instance, if you would envisage a patient that would have in a longitudinal measurement results like this, and you give the patient really insight into his or her results, then this patient may see, okay, this is what I normally should be between these two broken lines, and this is what my results look like. And this patient may then think, well, perhaps, yeah, it may be related to the fact that sometimes I may take my medication in the morning, sometimes pretty much at noon, or it could be early afternoon as well. Sometimes I take it before I have breakfast, sometimes after. And then if you make the patient co-responsible and give the patient insight into the results, then this patient may change the behavior. And actually, this may be what you get later on. And so this may be actually a way better control of the patient. So you don't have underdosing or overdosing, which may lead to adverse events. And you may have a patient which is better under control and which is better treated with exactly the same medication. Also, if we look at patient acceptance, and this is again from a study that we performed, and we asked the question whether patients considered the finger stick really painful, then the majority considered it not that painful at all. If the patients were asked if they considered the sampling quite easy, over 90% considered the sampling at home very easy to easy. And importantly, these were first-time users. User-friendliness, and this was in the context of volumetric absorptive microsampling, but other microsampling approaches may hold true for those as well. More than 90%, again, more than 95% actually, considered this very or moderately user-friendly. And then even if we asked if the patients had a preference when compared to monthly sampling, for instance, by a nurse or by a medical doctor, over 80% actually really preferred the microsampling. Importantly, it's not just the patients that matter, it's also the lab eventually that needs to monitor these samples that matters. Because we can say, okay, we have this flow of patient samples, but yeah, obviously these have to be monitored somewhere. And typically clinical labs at this moment in time are not really suited, are not really, don't really have the analyzers that have the appropriate throughput to work with these small samples, with these microsamples. Clinical labs are typically tailored to work with blood tubes and not with microsamples. And automation may mean an important way to push that field forward, and also costs, because if you have manual handling of samples, for instance, manual handling of microsamples, that means an additional cost to the patient, to the lab. But also, if you have devices and that cost more than blood tubes, that also may be something that a lab takes into account. So also these two things are important aspects that one needs to think of. In terms of automation, I genuinely believe that to have a real implementation in clinical labs, that automation is where we need to go, because it increases the throughput, it increases the safety, reduces the hands-on time, and also reduces the risk on human mistakes. At this point in time, there's different fully automated methodologies available for fully automated extraction of dried blood spots, so dried filtered paper. But if we consider these alternative devices, there's no real commercial systems available yet, or widely available yet, that allow fully automated analysis of such volumetric devices. So here are just some examples of systems that have been marketed or that are currently being marketed. And for instance, we have developed some applications for instance, antiepileptics, which are drugs which are present in rather high concentrations, but also more challenging application. For instance, if we think about immunosuppressants, and there we look really at low nanogram per milliliter analytes, and we could show that we could get trustworthy results in a fully automated manner. It's not just the analysis that I listed about antiepileptic drugs or immunosuppressants, but I could also think about antiretroviral drugs, also in toxicology context, newborn screening, vitamin analysis, like I already said, in diagnostics field, and so on. So there's a multitude of applications for which fully automated DBS-based analysis has already been demonstrated. And I can refer to the paper shown over here for an overview on this. Again, we already applied fully automated dried blood spot methods for therapeutic drug monitoring of immunosuppressants using real patients. And now actually we're also doing that for patient monitoring at home. So which is a field in which probably the clinical analysis of dried blood spot in the field of therapeutic drug monitoring is the most applied methodology. In addition, we should have realistic expectations. If you think about microsampling, this is just magic. We are not the wizards or the witch that can all of a sudden make things happen that otherwise aren't possible. We must have realistic expectations. It's not if something can't work with dried blood, with liquid microsampling, that dried blood microsamples may provide per definition the right answer. It's also like that for some clinical questions, microsampling may just not be the best answer. I already pointed out the fact that there may be improved stability in dried samples, for instance, but still, we just may decide that stability is not good enough in practice. And intrinsically, there will be some additional variation. And then the question is, what variation is still acceptable if you consider patient treatments? And how sure are we that we are currently, with conventional sampling, are measuring the right thing? Again, I refer to the fact that if we just have a single point in time and we measure a concentration in serum, plasma, or blood, to what extent are we sure that that concentration that's currently measured in a hospital environment is the representative concentration for the patient when considering the patient taking the medicine at home? So again, that's something that may define the acceptable variation. If we consider accelerating the uptake of dry blood microsampling, we can look at two different approaches, a bottom-up approach or top-down approach. And it could be shown over here. A bit a negative approach, as I see it, is the top-down approach, first shown here on the left. Top-down approach is like a policy change, and from now on, everyone has to do it in a certain way. A top-down could also be like just competition, because one lab has embarked upon microsampling, samples get sent via regular mail to that single lab, and other labs feel they are missing out, and so obviously also like that will implement microsampling. Otherwise, because they may lose the competition and they may not they may not get the samples anymore that they used to get. So these are two ways in which microsampling can be implemented. This is not really like the positive way if you have to do it or if you only do it because of competition. There's also what I would refer to as bottom-up approaches. And then it's up to us as researchers to show that it really can work. And if people can really see that it can work, or if politicians, for instance, can see that it can work, if financiers can see that it can work, then they may be really starting to push it forward as well. We should also better show that patients are capable of self-sampling, should show that it can provide reliable results, and also importantly show that there is economic benefits, because that's also very important, the economic benefits that maybe lie in the approach of microsampling. And with that, I've come to the last point of this list. In the end, and you could look at it from a negative or a positive point of view, but in the end, you could again consider it's all about the money. And like I already said, it's not necessarily a negative point of view that you should look at it. For instance, there's been a publication that really showed that if you look at it from a cost evaluation perspective, that dry blood microsampling at home as compared to conventional sampling for TDM in children was really cost beneficial. Obviously, there are several things that we need to take into account. There's the cost of the device. Like I already said, this could pose a hurdle. And also, who will pay the device and who will save on costs? And for instance, we could consider that the patient may pay to some extent for a device, because if the patient doesn't need to go to the hospital to just have a blood draw, then the patient may really opt, okay, I will pay $10, I will pay $20 to have a device that allows me to sample in the home environment, because I don't need to take a day off. I don't need to take my car to drive to the hospital, to wait there for a doctor to take my sample, and then drive back, be in traffic and so on. So that could be really something that the patient may opt for the home sampling. And very importantly, even perhaps more important, microsampling at home and getting better insight into the levels of medicines in patients may actually lead to better use of medicines that work. And some of these medicines are really expensive. And this could lead to improved compliance, reduced side effects, and actually really an improved outcome. And there are several initiatives that are now taking place, in which therapeutic drug monitoring, and could also imply therapeutic drug monitoring in the home environment, that really the improved outcome of therapeutic drug monitoring via microsampling could be shown. And if you can really show that we have a better outcome when using actually just the same medication in these patients, this could really demonstrate to the financiers, and this could be Social Security, for instance, that microsampling is really cost beneficial. So with this, I've come almost to my conclusion slides, and we could actually consider an interplay between different players. So here, importantly, in the center of this scheme is the patients. Patients should always be in the center. The patient is in contact with, for instance, the medical doctor. Patient is in contact with the pharmacist, and patients could send, for instance, a sample to the clinical lab. So we can consider the scenario in which the patient is in contact with the pharmacist, shown here at the bottom left, and the pharmacist may not just supply, for instance, the medication to the patient, but may also supply a self-sampling kit to the patient. The patient could take the medication, could self-sample at home, and then send the self-sample, the dry blood microsample, for instance, to the clinical lab. The clinical lab reports to the physician, shown here on the top of the slides, and the physician can then be in contact with the patient and say to the patient, there's no need that you come for an appointment, because I can see from your blood values that everything's under control. Alternatively, the physician could also say to the patient, okay, I've really seen there's something wrong on your blood levels, because we know that longitudinally you're typically quite well in control, but I've seen now that your immunosuppressant levels are way different than they used to be, And I can also see that your creatinine levels, which were used also in the same sample, that they're off. So you should really come and see me. And so this actually may save time for the physician to really orient her or his attention to those patients that really need it, as you may have a better patient follow-up. So to conclude, dry blood spots are just microsampling in general, and analysis has evolved a lot since the establishment of newborn screening. Microsampling or dry blood spot sampling offers a wide variety of advantages. There are several issues associated with microsampling and DBS sampling, and analysis and validation may require actually substantial extra efforts. But again, there are some guidance documents that should help in that matter. The last few years, there's a wide variety of approaches that has become available that allow to deal with the issues and challenge. And now, microsampling is increasingly being used in a wide variety of applications. And for instance, if you look at therapeutic drug monitoring and toxicology, it is already being used. Important steps have been taken and continue to be taken towards implementation in routine. For instance, if I just take in the context of my lab, we've set up robust procedures and have routinely implemented this. For instance, we have an ISO-accredited procedure for drug of abuse, for GHB, but also for alcohol marker phosphatidylethanol. And for instance, this phosphatidylethanol determination, we have it in routine use for driving license regrouting cases. So we get samples from all over Belgium, but also actually from abroad, dry blood microsamples, and we do currently thousands of these analyses on a yearly basis. And in conclusion, things work, but we do still need more real-life data, as well as studies demonstrating the clinical, but also importantly, the financial benefits of microsampling. And with this, I've come to the end of my presentation. I wish to thank my team, current team is shown over here, but also the past team. And I wish to thank you for your attention. I'm open for questions. Thank you.
Thank you, Christoph. Wonderful presentation as ever, and bang on time. So we do have some questions. I'm just going to take them in the order that they've come through. The first one's from Russ, who congratulates you on your talk. And he asks, are there considerations you could comment on for the potential issues of kit costs plus two-way shipping sent to the patient's homes? These may not always being covered by managed care, insurance, or government entities, so raise out of pocket for the patient. He says, for consideration, perhaps the most important omics is the economics, which is impacting our ability to be further forward. So I guess he's asking, what can we do about getting past these perceptions of extra costs?
Yeah, it's a very relevant point. And like I said in my presentation, unfortunately, sometimes in the end, you could say it's all about the money, it's all about costs and who saves and who has to pay for some things. And actually, earlier this week, I had a discussion also on that with some clinical labs. And actually, what came down from that is that clinical labs don't want to pay for these extra costs. They're very cost sensitive, and you can easily envisage a scenario in which actually the costs are shared. Because anyway, if you have a dedicated device that really allows you to have trustworthy sampling at home, in which you can get a good quality sample, it obviously comes with the costs. Mm-hmm. And you can easily envisage a scenario in which you have a shared let's say, coverage of that costs. Part of that could be covered by the clinical lab because they may have an advantage that they don't need a nurse that is required for the sampling. Part of the cost could be covered by Social Security because if you can have a better patient follow-up, in the end, Social Security could benefit from that. And part of the cost could actually also be covered by the patient because if the patient, like I said, doesn't need to make the move to the lab or to the medical doctor's office to get the sample taken, then that's really an advantage for the patient as well. And I do think that quite a number of patients would be willing to pay somewhat for that, just for the comfort of being able to sample in their home environment.
Okay, thank you, Christoph. We'll go to the next question from Elia. What is regarded as acceptable variation for measuring small molecule metabolites when using volume-controlled substrates? Example, Capitana B10.
Yeah, it's a challenging question, and it's the answer will largely differ depending on to whom you ask this question. Clinicians and also if you think about people that are working in typical clinical labs will really focus on what the conventional kits are currently capable of doing, and those have really low CVs, so really low imprecisions. Typically, if we look at mass spec-based procedures, if we look then also at microsampling and microsampling combined with mass spec-based procedures, we will have a bit a larger variation. Typically, I would consider everything which is below 20% as a total imprecision as acceptable. But I am aware that in several instances this may not be considered acceptable. And the reason why this may not be considered acceptable is, so for instance, if you think about therapeutic drug monitoring, that clinicians may really, like I already said, focus on what they are used to, the variations of actually the instrumental variation. And they just simply disregard the variation that takes place in the patients. And like I already said, if you would have the patient come in today and tomorrow and the day after and the day after again, and you have a clinical sample taken, they would be really, they would be offended, as to say, by the variation that they would see in that patient, in which they would actually expect the same result to take place based on the fact that this patient is supposed to take the medication in exactly the same manner. And so this is something which is largely disregarded. And so I do think that we can allow some larger variation than, for instance, the 5 or 10% that they would typically focus on. So we can extend that to roughly like 20%.
Yeah. I think we lost your microphone at the end there, Christoph. Hopefully.
Oh, I'm sorry. So I think we can extend that a bit from what originally is like 5 to 10% to roughly 20%.
Thank you. We have a question from Vincent. Can you comment on how standardized you think fingerstick sampling really is in practice, considering known capillary effects of milking one's finger or lysis of cells?
In our hands, we've obtained really good results in terms of reliability of microsampling, provided that patients are instructed properly. So what we typically do is instruct patients to wipe off the first drop of blood, and then if we then look at repeated samplings, so 2 consecutive drops of that patient, and even also compare it with venous results, then we can get results which are really very consistent. So we've demonstrated that for several analytes. We demonstrated that, for instance, for this alcohol marker PET, but now recently we also demonstrated that for tacrolimus, so using a volumetric device, where actually the results were even better than I expected them to be. And so I do expect that just in a few years from now, we may see more widespread application because it can work really, really well.
Yeah. Thank you, Christoph. Next question's from Marlene. Do we need reference ranges for clinical assays to be established for microsampling assay if the idea is that we get similar results from blood plasma serum?
There's no easy answer to that question. It's a very relevant question. In a sense, my view on it is if there's a wide variation on the blood plasma ratio, then we should not try to convert a blood concentration to plasma concentration. We do all these efforts in order to get a really trustworthy concentration in blood, in which we really know that what we get is correct. And then it makes no sense, in my opinion, to try to convert that blood concentration to plasma concentration if we know that we just will introduce solely by that conversion of a factor of 20, 30, 40% uncertainty. And so in that perspective, I don't think it makes sense to force the conversion. And obviously that may imply that you may be confronted with plasma reference ranges and blood reference ranges, which is also not really practical, which may lead to mistakes. But there's also other examples where clinicians need to take into account different ranges depending on the matrix, and it also works. So just saying per definition that having a blood reference range versus a plasma reference range is not possible, is I would say underestimating the clinician's capabilities.
Yeah, I think that's a really good point. Next question's from Ross, and it's another complicated one, I think, and another one that's very pertinent at the moment. What steps need to be undertaken and by whom to implement dry blood spot workflows under IVDR, FDA, and other governmental jurisdictions? So obviously there's a lot of discussions going on with the FDA at the moment. Just some of your commentary around that.
Yeah, very relevant question, very timely question as well. In a sense, I don't consider microsampling that much different from regular blood samples in this perspective, because it's the issue primarily deals with lab developed tests, and it's perfectly possible if you have a kit that works in a certain context for just regular blood samples or regular plasma samples, regular serum samples. It may be very well possible to apply that kit to a sample which is a dry blood microsample. So I don't think the sample doesn't matter necessarily that much, because if you consider a dry blood microsample as a blood sample, a form of a blood sample, rather than a liquid form, it is a dried form, it's just part of the sample preparation, then it really shouldn't matter. And actually what we do, I even participate to proficiency testing schemes where actually the results are reported in serum concentrations, and where we do the analysis on dry blood microsamples, we convert them to serum, and then we report as serum, and we have really good results in these proficiency testing schemes. So even although there's no proficiency testing available for blood, and even not for, and obviously then not for dry blood, with our approach, which we do the analysis in dry blood, we can report as serum, and we pass these proficiency testing schemes. So it is possible to really get trustworthy results and even participate to proficiency testing schemes. And the IVDR regulation is primarily about lab-developed tests, and I think there's ways to fit also microsampling in that perspective.
Thank you, Christoph. I think we've got an interesting time ahead of us. We've obviously got the FDA webinar at the end of the month, telling us what their plans are. The next question is from Vincent, which is basically covering the same territory. So, Vincent, if you don't mind, I'm going to skip over that one unless there's something I've missed there, and please post that again. Etienne just said, wonderful talk. Nicholas says, according to you, which is the main obstacle today for implementing microsampling at home in the general population in Europe? How can we convince end users and health professionals to do the switch from the conventional vial?
Main obstacle, I would say, to get it really implemented in clinical routine is, getting the finances streamlined and who will pay for what. I think that's one of the key things. That's one. The second is labs, like I said, labs need to be ready for it. If you now say to just a standard clinical lab in hospital, Can I send you microsamples, and can you handle those? Most labs will say no. Typical clinical labs are focused on automation. If I think about clinical lab here in Ghent University Hospital, if a sample, a blood tube, is taken in the emergency departments, it's transferred via tube system to the clinical lab. There, it automatically is sorted into a rack. It automatically goes to the analyzer, and within less than an hour, the result is presented at the emergency departments. Not a single person in the clinical lab has touched the tube.
Yeah.
So in other words, everything is fully automated. At this point, we are not there yet for dry blood microsampling, and clinical labs don't want to have more people in their lab. They don't want to have more lab technicians. They want to have less. They want things to be automated. They want things to be working with robots, because robots can work 24/7. And so robots don't make mistakes if they're properly programmed. And so that's one very important aspect from the analysis point of view. And apart from that, like I said, the cost perspective, who will pay for what?
Yeah, I think it's interesting. Hopefully, the EU-funded IHI initiative, looking at the logistics of home blood sampling across Europe, is going to be interesting in helping us to understand the challenges and how to overcome them for this kind of thing as well. I think it's a really exciting initiative.
Yes, indeed. And like I said, also by demonstrating that it can work, things can really start. If I consider the context in Belgium, we were amongst the first ones to launch, let's say, several projects based on dry blood microsampling. And then if I look now, there's so many things going on. We have a population study going in the context of a food consumption survey. You have a population study to look at environmental pollutants, which is based on dry blood microsampling. So there's really many things going on. In a routine context, we use dry blood microsamples, and we on a daily basis get dry blood microsamples via the posts. So FedEx sort of has almost a hub here in our lab, and by bringing on a daily basis dry blood microsamples over here. So that really shows that things are taking off, and by showing that it can work and that you can in that way provide a very good and reliable service. That also helps to move things forwards. And like that, other people may start wondering, well, why am I not doing this? And then if more and more start doing that, then also the ones that are in charge of let's say making the decisions about finances, they also may wonder, okay, why do we pay, for instance here in Belgium, patients, cancer patients can get a free taxi to the clinical or to the hospital, for instance, for blood draw. Obviously, you could wonder that also costs money. Why invest money in a taxi service if you can just have that patient sample the sample, collect the sample at home? So those are all things that you need to show that can work, that you can get reliable results, and that you can have even better patient follow-up than currently is taking place.
I think that's a wonderful example, Christoph. There are a few more questions, but we now run out of time. I'd just like to thank you, Christoph, for an excellent presentation, for being a leader in this field, for sharing your expertise, and showing with your laboratory that this can be done routinely. You're not just operating an academic lab that does some nice academic publications. You are actually doing this day in, day out for real samples that are making a difference. So just before we go, I'd just like to put down my own. Yeah, okay. So just this webinar and a series of webinars brought to you by the Patient-Centric Sampling Interest Group. I'm not going to really read this out, but if you want to learn more, the QR codes on the right will take you to our website and to our LinkedIn group. We've just hit 300 members for our LinkedIn group. We can only operate with members going through activities that really help push forward and break down these barriers that Christoph was talking about, and working together across organizational boundaries. And just finally, I'd like to thank our partners and our sponsors who make these events possible. And with that, I'd like to thank you all for your attendance, and thank you again, Christoph, for a wonderful presentation and being a leader in this field. Thank you all.
Thank you.